So what I'm working on right now is challenging the idea that cancer cells are a single entity, a single type of bad cell. And so I study blood cancer, and what we find is that when you look at thousands of cancer cells, that they differ from one another. There's different types of cells present even in the same person's cancer. And so it turns out that these different types of cells resemble different stages of normal blood development, and that they're sensitive to different types of drugs. And so the idea that thinking about the recipe of each person's cancer, what types of cells they have, and using that to start to predict what drugs they might be able to benefit from. We've been trying to move from these deep portraits of cancer cells in individual patients using, multi-omic data and going into patient cohorts with hundreds or thousands of patients to start to predict survival rates and drug responses. And so what we're hoping to do is use this as a tool to think about what trials patients should be prioritized for based on the sensitivity of the cells that they have to these various drugs. So ultimately, the goal is to personalize patient treatment for leukemia. I started in cancer research at age 18 throughout my undergrad, and it was really academically interesting. But I think a really formative moment for me was when I flew back to Beijing for 48 hours to say goodbye to my grandmother, who was dying of terminal colon cancer. And so that was really when it changed from becoming an academic exercise into something that was much more personal and human for me. I'm now back in medical school, and I have, after this, 2 more years before residency. But my hope is to become a clinician scientist and spend my time split between doing research to think about new treatments for cancer and then also treating patients.